Good morning to all. My name is R. Maniyandan. My topic name is vectors. Characters of vectors: self-replicating multiple copies, replication origin site, cloning site, selectable marker gene, low molecular weight, essentially isolated and purifies, essentially isolates into host cells. அடுத்து பார்க்க போகிறது the molecular cloning process involves the following steps. cloning process என்னென்ன ஸ்டெப்பு ஃபாலோ பண்ணுறது அப்படின்னு தான் பார்க்க போகிறோம். அது ஃபஸ்ட்டு ஸ்டெப் ரெஸ்ட்ரிக்ஷன் என்சைம்ஸ். இது ஹெல்ப் இன்டு கட் த டிஎன்ஏ இன்ட்ரெஸ்ட். Ligation. DNA of interest is fastered to the vector DNA with the help of ligase. Next, transformation helps into the introduction of vector DNA into the host cell, often by bacteria or yeast. Then the host cell copy the vector DNA along with their own DNA, creating multiple copies of interest DNA. Next, isolation. The vector DNA is then isolated separate from the host cell and then purified. Next, classification of vectors. Cloning vectors used to clone virgin DNA. Artificial vectors. There are generated vectors so that any foreign DNA can be transferred in RNA and translate into proteins. Importance factors of cloning vectors: ability to replicate into host cell, and restriction enzyme sites for intentional cloning, generate marker to select for host cells containing the vectors, low molecular weight. This is the structure. Types of vectors. First step, plasmids. Extrachromosomal double-stranded DNA molecule, curricular cell replicating, contain origin of replication, allowing for replication, independent and found in prokaryotes. Classification is plasmid. Fertilized plasmid. Fertilized plasmid is also known as F plasmids. Contains transformed gene that allows genes to be transformed from one cell to another thought concentration. Example, F plasmid of plasmid of Escherichia coli. B. Coil plasmid. Coil plasmid contains genes that make bacteriocins, also known as colchians, which are proteins that kills the bacteria and thus define the host bacterium. Example, Escherichia coli. D. Decorative plasmid. Decorative plasmid help the host bacterium to digest compounds that are commonly found in nature. Chooses chaper, xylene, toluene. Those plasmids contain the gene for special enzymes that break down specific compound. Example: toll of Pseudomonas putida. Next, virulence plasmid. When a virulence plasmid is inside the bacterium, it turns that plasmidium in pathogen, which is an agent of antigens. Example: type plasmid of Agrobacterium tumefaciens. Next, bacteriophage lambda. Phage lambda is bacteriophage or phage is bacterial virus that uses E. coli as host cell. Its structure is as a typical phage head tail fiber. Lambda viral genome contains 485 kb linear DNA with 12 base ssDNA stocky end at both ends and end is complementary is sequence and can hybrids to each other. Example: phage lambda M13 pages. Next, cosmid. The cosmid vectors are the combination of plasmid vectors and the cos site which allows the target DNA to be inserted into the lambda head. Example: PJB8. Artificial chromosome. Artificial chromosomes are DNA molecule or formed assembled in vitro from defined constants which generate stable maintenance of large DNA fragments with the properties of neutral chromosome useful for genome sequencing program functional characters of entire genome region and for translation of large DNA segment into human and non-human mammalian cells. Types of artificial chromosome. A. Bacterial artificial chromosome BAC. B. Yeast artificial chromosome YAC. Thank you for all.